Okay, hello everyone. I am Hamid. I'm a PhD student at the Oslo University Hospital at the Intervention Center and also at the Faculty of Medicine at the University of Oslo. I'd like to present our paper titled Anomalous Diffusion of Extracellular Vesicles in an Extracellular Matrix for Molecular Communication. Okay, so recent COVID outbreaks actually reminded us that new treatment methods can help us to solve new problems that we face in life. So one of these nice solutions was the discovery of this COVID vaccine, which was based on RNA. So we found that like drug delivery methods can help us. So to study more on these drug delivery systems, we can use like molecular communication theory and study actually the cell-to-cell communication. So to do this, we need to first study the cell-to-cell communication based on like this. For example, we can see this figure here. So basically, there are like the donor cell and the target cells. So these two types of cells can also send and receive the chemical signals at the same time, which can be considered as transmitters. So basically, the donor cells can release the different types of chemical signals, such as proteins, lipids, or different types of vesicles, which we are going to consider a specific type of vesicles in our study. And these chemical signals, they go through and diffuse in different types of media inside the body, such as the like circulatory system, such as blood vessels, or inside the extracellular matrix, which are a specific region inside the tissue. And then these chemical signals can be picked up at the target cells, and they can change the functions of the target cells. So we wanted to study the extracellular vesicles. So they are seen to be so-called Warwick. So they have some specific features that are very interesting for us to use in the molecular communication. The first feature is that they can carry different types of molecules, such as proteins, lipids, and also genetic data, such as RNA and DNA. They can also target specific types of cells or the target cells through these membrane proteins. And also they can be engineered to carry such therapeutic and particle drugs for the treatment methods. So our aim is like studying specifically these EVs, extracellular vesicles, in different body locations, which in our study we specifically use the cardiac tissue and the whole cardiac extracellular matrix for our study, because one of the leading disorders in the world is the cardiovascular disease. So to study the actually diffusion of EVs inside the cardiac tissue, we first consider the cardiac space. So basically, cardiac space is comprised of different types of cells, such as cardiomyocytes, lymphocytes, T cells, and so on. And these cells are attached using the cardiac extracellular matrix. So the cardiac extracellular matrix is mainly based on collagen type 1 and collagen type 3. And these collagens, for example, here we can see the three types of cardiac ECM, such as deep elastic ECM, soft stress relaxing ECM, and stiff stress relaxing ECM. So in these different kind of regimes, the matrix mesh are different. And so there is one question that the diameter of this cardiac ECM mesh is less than the diameter of EVs. So the question how these EVs can propagate and diffuse inside the cardiac ECM. So the answer is because of the specific features of EVs EVs that made us interested to study these EVs, these actual vesicles. So in our model, we actually modeled the diffusion and propagation of extracellular vesicles using a three-dimensional partial differential equation with an initial condition. So we have four basic parts for our modeling. So at the first part, we have the volume source function in our 3D PD. So compared to the works in the literature, the source function in our work is not a point source. It has a volume, which is different and more practical because in nature, so we don't have like the point source. It's better to use a volume source function in our modeling. Another important contribution in this work was time-dependent degradation and modeling, and considering this in our modeling, and also considering this in our analytical solution. So in the time-dependent degradation, we considered two important features as the extracellular binding of EVs to the matrix mesh, matrix actually molecules, and also the half-life of EVs, which I elaborate more on this part in what follows. Also, we considered the unique ECM modeling, which are tortuosity and volume fraction of the cardiac ECM. So basically, tortuosity models the hindrance of the ECM, that how these ECM are kind of limiting the distribution and the movement of EVs inside the cardiac ECM, and also the volume fraction models the accessible space for EVs to propagate, considering the whole volume of the cardiac ECM. And the important part of this modeling was modeling the anomalous diffusion, which was a overload function. And I will elaborate more on this in next slide. So the biodistribution modeling of EVs in the cardiac ECM has a time-dependent degradation. So the time-dependent degradation has two parts. The first part is the extracellular binding of EVs to the matrix molecules. So so EVs have like some membrane proteins that can be kind of detected by the other matrix molecules, and then they can be binded together. And these can actually degrade the whole concentration of EVs in our medium, and actually avoid them to reach the target cell or to the viewpoint or the distance that we want to. Another thing that we consider is the half-life of EVs. So basically we have the half-life of EVs in the whole body. So for example, wet lab, the half-life of EVs is around 6 hours to 12 hours, but inside the body, it's not like this. And inside the body, we have the half-life from 2 minutes to half an hour. It's not a long, time. So we need to consider this in our modeling too. So for the anomalous diffusion, so we considered an overload function for the modeling of anomalous diffusion. So basically, the Brownian motion, actually, which can be the normal diffusion of EVs, it's not tractable. It's not true modeling for the distribution of EVs inside the cardiac ECM. So because of that, we consider an overload function for modeling the anomalous diffusion. So here also we are considering these three regimes which have been experimentally proven in the literature. So for example, these three regimes also can be possible to model different types of disease. For example, a steep elastic ECM is modeling the accumulation of excessive ECM like matrix molecules inside the ECM. And it can be kind of show the story dysfunction, which is a cardiac disorder for the heart tissue. So another interesting thing about the modeling that has been given in the paper is that the boundary condition, we considered a 3D environment, but I think is using the auxiliary function in our modeling, it's possible to use different types of boundary conditions such as Newman or Robin boundary condition. So I don't want to go to details for the mathematical formulation inside the lecture, and but I would like to present some of our results here. So we actually solved the 3D PV given in the lecture using the Green's function, and to prove it, in the revised version we provided the particle-based simulation. So here we can see that the particle-based simulation, the PBS simulation, actually is verifying our analytical solution. And here we are showing the different anomalous regimes, such as steecholastic, soft stress relaxing, and steechostress relaxing, and also the normal diffusion, and also the PBS. So one interesting feature in our modeling is the degradation. So the degradation can make a lot of changes and can change the whole thing actually. So here, for example, we are showing the concentration of EVs in same location as 3 microliter from the source function using 2 different types of degradation rates. Here, the degradation rate of the figure A is much lower than figure B. Here also we can see that the steecholastic concentration, concentration of EVs considering the steecholastic is more than normal diffusion. But here, the concentration of the steecholastic is much lower than the normal diffusion. But the locations are same. So here we can actually understand that there's, by considering the subdiffusive environment, or we can say anomalous diffusion in cardiac system, the whole scenario will change, and we cannot say that always, for example, using steecholastic is always greater or lower than the normal diffusion or other types of subdiffusion. And also we can say that there is balance. There is a balance between the degradation that we have in the environment and also the location of the dew point. So in another simulation, we are simulating the concentration of EVs in a more longer distance than the previous ones, which is 5 microliter. So if you remember, in the last slide, the concentration of the EVs was the steecholastic scheme was greater than the normal diffusion, but here the normal diffusion is more than the steecholastic. But still, in the having this degradation, the steecholastic has less than the normal diffusion. One reason for this is that the EVs in the steecholastic regime, and actually they tend to less move in the environment. So because of that, in long distances compared to the short distances in the last slide, they tend to move less in the long distance, and they probably tend to stay at the short distances. And because of that, we can see more of EVs, we can see more concentration of EVs at the short distance. So in this slide, I would like to show some of our simulations about the concentration of EVs in different dimensions, X, Y, and Z, with respect to time. So if you notice that, so we have 2 things in all of these 3D results, and release rate at the source function, and actually can prove that the simulation and the whole analytical solution is true. And also another conclusion that we can have from these three figures is that they all seem to be same, and the reasons it's because we don't have any velocity in the cardiac ACM. If you have velocity in different directions, the whole the 3D actually illustration they will change. So at the end, I would like to conclude this letter. So in this work, we actually modeled the anomalicity of cardiac ECM for the propagation and the movement of EVs, which can be used in the future works and probably future applications to develop new types of direct delivery systems. And I would like to thank you all of my colleagues. Some of them are not in this picture, but it was the recent seminar that we had in Norway. And yeah, I'd like to thank everyone that helped us to make this paper. Thank you.
Thank you, Hamid, for the interesting presentation. Any questions?
So where you compare your results to particle-based simulations? Yes, 8, perfect. Yeah.
8.
Yes, slide
Okay.
8.
Yes.
There seems to be a mismatch between the particle simulations and the analytical model.
Yeah, for increasing times, so around 14 to 20.
Yeah. Actually, this is because of the computational, the whole computation in the particle-based simulation, and actually we increased the number of repetition for our previous, but still we have some mismatch, and so actually we mentioned this in the revised version of the letter, that why we have this this mismatch, but it was not possible to kind of be exactly same. But I can say that it's very fine, the whole analytical solution.
Okay, thanks.
Okay, thank you. Hamid, I have a question? So the particle-based simulation, what simulation framework have you used for that?
We used actually the particle-based simulation in C, and so we generated different mesh, and then using the power law function that we have for the subdiffusion, we modeled the whole simulation, and also at the source function we considered the Gaussian the function for the source, for the volume source function. So in this case, we modeled the source function in the space, and also we considered this function here as the release rate of EVs.
Okay, thank you.
In the time.
Any other questions?
Yeah.
Okay, thanks a lot, Hamid, for the presentation. Thank you.